right, got it. We are being live.
That's cool.
It's moving to YouTube.
Technology at its finest.
I love it. Once I learned how to do this, I was like, wow, this is not so bad. Great, got it.
I might be asking you.
Moving to YouTube. To mute this.
Technology at its finest.
I love it. Once I learned how to do this, I was like, wow, this is not so bad. Great, got it.
I might be asking you.
Moving to YouTube. I need to, you guys, I gotta mute that.
Technology at its finest.
I love it. Once I learned how to do this, I was like, wow.
Where is that coming from?
Hang on a minute. It's
I might be asking you.
it is. I have to stop sharing my screen. You guys, I gotta mute that.
Technology at its finest.
I love it. Once I learned how to do this, I was like, wow.
Where is that coming from?
Hang on a minute. It's it is. I have to stop sharing my screen. You guys, I gotta mute that. We're solid.
Okay. Whoa.
Yeah, it was just echoing itself. It was pretty great. That's why I just muted my microphone and let it work itself out. We're good.
I couldn't find it for a moment, so it's beginning to panic. Okay.
Understandable. No
Hey.
worries.
Hey everybody, welcome. For those of you that are in our virtual science colloquium meeting space, I want to welcome you. I'm Elizabeth McInnes and we have Denise Godbout-Avant. We're both part of the Science Colloquium Committee hosting today's event. And today we have Dr. Kari Arnold. And she's going to give us a great talk on plant pathology, past, present, and future. Before I hand this over to Denise for introductions, I do want to let you guys know that on October 13th, we have Greg Stock, park geologist from Yosemite National Park. Really excited about this talk, The Rise and Recent Fall of Sierra Nevada Glaciers. And in his talk, he'll be talking a little bit about how climate change has impacted the glaciers of the Sierra Nevada. So I'm gonna hand it over to you now, Denise.
Good afternoon, everybody. It is my pleasure to be able to introduce Dr. Kari Arnold, who I had the opportunity to work with in the Master Gardener program. And she was born in Sioux Falls, South Dakota, and grew up in Omaha, Nebraska. She graduated from the University of Nebraska with a bachelor's in science and horticulture and plant science. She then moved to Sacramento and got her master's in science and PhD in plant pathology at UC Davis. And she studied viruses and grapevines. And viruses, of course, are obviously very appropriate these days, even in grapes. Her career has been currently at the University of California Cooperative Extension Orchard and Vineyard Systems Advisor in Santa Claus County, which is a bit of a mouthful. And her interests include science, gay, kayaking, hiking, gardening, mortgage, and singing, writing, music, which I thought was really interesting in a rock band. So we got science and art together, which is cool. And I'm going to hand it over to you now, Kari.
Well, thank you very much for that excellent introduction, Denise. And thank you, Elizabeth, for having me today. Thank you for the invite to both of you. And thank you to all of those attending. I hope I don't put you to sleep. I'll try to keep this rather entertaining while also talking about some very interesting topics in plant pathology and in science. So with that, we will move on. So yes, I would like to quickly define plant pathology and some basic concepts so that we kind of fully understand what we're talking about here. discuss the disease triangle and the disease cycles, review causes of plant disease. We can discuss how to prevent, control, and manage disease, discuss some hot topics, and look to the future. So generally speaking, what is plant pathology? Well, it's the science of plant diseases. So sometimes people are like, so are you a plant doctor? And I'm like, yeah, kind of. It's pretty cool. So there are up to as much as $9 billion in crop loss to plant diseases in the world. And plant pathologists study diseases, abiotic plant disorders that mimic disease, and insects that carry disease as well, because insects can carry disease from plant to plant. Pathogens, generally speaking, may interfere with photosynthesis. They may interfere with water absorption, infect and plug up the vascular tissue. They can increase respiration rates in infected cells. They can alter the permeability of cell membranes. They can alter protein synthesis, and they can disrupt metabolism of genetic materials. We're going to talk briefly about a couple of historic disease cases, because this is kind of like where plant pathology finds its place and where it grows from. A few examples that we'll dive into are St. Anthony's fire, the potato famine, coffee rust, Dutch elm disease, and actually Huanglong being otherwise known as citrus greening in citrus. So we're going to jump right into the potato famine. So in the 1840s in Ireland, they historically stored potatoes underground, or at least in these sort of cooler storage areas, and they were kept sort of, it was moist, sometimes there was water in there. So when they would harvest the potatoes, they would put all of these potatoes into these storage facilities that were cool and moist. And after a number of years of doing this, year after year after year, they go in to pull from their harvest stores, and they pull out nothing but black mush. And they're like, oh my gosh, what's going on? And then they try to grow potatoes, and they also fail at growing the crops. So they're like, what is going on? And this is this is middle of the 1800s. We have no idea what pathogens are. We have no idea what plant pathology is. It's just black mush. So 1 million died, 2.1 million left or fled the country because of starvation. Quite frankly, there was no food. And it was the perfect storm. The nation, unfortunately, the nation was dependent on a susceptible genotype or host. It was a conducive environment. We had moist, cool conditions. And there's this possibility that the inoculum, that the pathogen was introduced to the area. So the pathogen Phytophthora that we're going to talk about here shortly, the pathogen was not actually present in Ireland. It was likely introduced from elsewhere. So way back in the day, they had no idea what was going on. They're like, what is causing this? They believed in spontaneous generation. So living organisms generate from inanimate objects or rotting organisms. So, they just they believed that mold just showed up. They didn't understand that it was an organism growing and that it came from a spore. They didn't understand any of that. Oftentimes, and especially in this case, was it God's wrath? Is God angry at us? That's a possibility. Why is this happening to us? Is this an act by the devil himself? They actually sprinkled holy water on fields. They raced trains, discharging electricity to chase out the devil. They did everything that they could that they thought would be effective to save themselves from starvation and to save their family members and the people that they loved. And they didn't understand what was going on. So Heinrich Anton de Bari in 1861 decides to go out and pull some infected potatoes. And he takes a paintbrush and he touches the paintbrush to that white, strange material there. And he transfers it over to a healthy potato. And the craziest thing happens. Something grows. So what he's just done, and what is later developed as a methodology, is Koch's postulates. This was developed by Robert Koch and it proves causation. So certain steps must be taken to show that a certain microorganism is the cause of an infectious disease. And that's exactly what Anton did. He took that strange white material and moved it over to a healthy potato to see if he would see symptoms or signs of the disease similar to the previous infected plant. So this also brings up kind of a topical question of, does HIV cause AIDS? So one could argue that we cannot actually infect a human being with HIV to show that that person would develop AIDS. But there have been so many correlations and regressions to show that HIV is likely the cause of AIDS, because you get one and then you get the other. So that's sort of where that, every now and then you might hear somebody say, well, we don't have causation. Like, well, in that case, we have a lot of correlations and we can't just go around infecting people with terrible diseases. So that's where, if you hear something controversial along those lines, that might be why. But we have plenty of correlations to show what we cannot show by causation. So as for late blighted potato, I'd like to take this out and talk about the disease triangle, because the disease triangle in plant pathology, you have to have a causal organism, you have to have the environment, and you have to have a host, a susceptible host. Otherwise, you don't have a disease. You have to have all three, or you have nothing. You have a healthy plant, or you have an abiotic issue, so an issue not related to a pathogen. So with late blighted potato, we have, in Ireland, we have cool, damp, foggy conditions, generally speaking, and also in terms of storage. We have the common potato, which is clonally propagated. So if you've ever gone to a store and purchased seed potatoes, it's literally chunks of potatoes with roots coming out of them. So that would be a clonally propagated plant or crop. They're stored underground where it's cool and damp, and this is the current and future food source. So genetically speaking, it's the same plant being planted over and over again, stored, then replanted, stored, eaten, replanted, etc. And then Phytophthora infestans, which is the causal agent for late blight of potato. In Latin, they basically called it the plant destroyer, and it originated in Mexico and was introduced to Ireland. So what happened, the potato famine, the Irish potato famine that caused 1 million people to die and 2.1 million people to flee, is likely related to the introduction of a pathogen when some material came across the sea or across the ocean. This happens quite often. So you can see how vulnerable we might be as a human population when it comes to diseases. So as I said before, with the disease triangle, you have the environment, the host plant, and the pathogen, and that brings about the disease. So as for disease cycles, every organism has a cycle. Organisms have generations. So with that, the disease cycle for late blighted potato, you initially have inoculation. So a sporangium emerges and floats over to a susceptible host, lands on that host becomes inoculated. You have this penetration where the organism is penetrating into the plant tissue. That's infection. You have infection. Now the organism, the pathogen, is growing inside the plant tissue, and now the organism wants to reproduce and fly out to other plants. So that's where you would have your dissemination. And that can be in various forms. You can have sexual or asexual sporulation and reproduction, and it really just depends on the pathogen. So in terms of St. Anthony's fire, this was a really interesting excerpt that I took from National Geographic. So the 17th century French historian Francois Ude de Mézeray chronicled a plague that swept through southern France in the 10th century. The afflicted thronged to the churches and invoked the saints. The cries of those in pain and the shedding of burned up limbs alike excited pity. The stench of rotten flesh was unbearable. Throughout the Middle Ages, many outbreaks occurred, some taking tens of thousands of lives. Symptoms included convulsions, hallucinations, and excruciating burning sensations in the limbs. Dubbed ignis sacer, or sacer, I'm probably saying that wrong, or holy fire, the affliction blackened the limbs until they fell off the joint. This is still an issue. This is a disease of wheat, and it's called ergot of rye. It's caused by Claviceps purpurea. It's not religious in nature, but it is in the world today. It's nasty. You don't want it. There's a lot of ways that we work on controlling the disease so that people don't have it, because it is so awful when you're ingesting this toxin. And so what's happening is the fungal pathogen is producing alkaloids or mycotoxins. And those mycotoxins are actually leading to the illnesses that we just discussed. And that they were awful. And of course, at the time, they had no idea what was going on. And it was really sad because people would just die in the streets. But now we can control it because we understand the disease triangle. We understand that it needs a temperate climate. It requires cold periods for sclerotial germination. If you want to know more about sclerotia, feel free to look it up. Rainfall and high soil moisture. Rye and other grasses is the susceptible host. Oats, barley, wheat, things that we eat. And Claviceps purpurea is the causal agent. Creates these toxic alkaloids and causes ergotism in humans. And of course, we are fortunate not to have these issues given the supply chain of our food source in the country that we live. Sometimes you might see issues like this in livestock, because maybe they're eating grain that didn't go through the same level of selection and management to keep that out. So currently for ergot management, they use clean seed. So growers will grow clean seed. They plant clean certified seed. They ensure uniform stands. They constantly scout. They're doing sanitation of that site, crop rotation, deep plowing to bury the ergots, which is the blackened seeds. They use certain harvesting techniques, and like I said, scouting, field burning, chemical control, and genetic resistance. So they're trying to come at it from all various points of possibilities because we don't want to ingest this stuff. So that's an interesting historical disease to think about. Coffee rust is also a really fun one to think about. Historically, and it's because of the cultural shift. Historically, England drank coffee, and I don't know if anybody listening knows that because a lot of them drink tea now. So Ceylon, the center of production, exported nearly, my camera's blocking it, nearly 100 million pounds a year, mostly to England. In 1867, there was a reported coffee leaf disease, and 20 to 25 years later, the coffee industry was decimated. British planters started cultivating tea, and now England drinks tea. So coffee rust later entered the Americas in the 1970s, and there's still lots of opportunity for research and development in trying to figure out how to handle coffee rust. So it's still a problem today. But it's just crazy to see how it would change, generally speaking, what an entire country would drink simply because of the disease. That just affected, that actually didn't affect the people drinking it. It just reduced the supply. It just completely cut out coffee production. That was it. Now, something that's much more current is Huanglongbing, or HLB. And this is also called citrus greening, because as you can see, the fruit, they don't come to full maturation. They don't fully color up. You can see in this photo, half the fruit is green and half the fruit is orange. So it started getting called citrus greening. But they've changed that because the citrus doesn't really turn green. It's already green. It just doesn't fully ripen, etc. The fruit is gross. It's bitter. It doesn't taste good. This disease is caused by bacteria, and the bacteria reside inside the plant, inside the vascular tissue. And this bacterium is transmitted by a vector, so moved around by another organism. And in this case, it is the Asian citrus psyllid. So unfortunately, the Asian citrus psyllid was introduced to California not too long ago, and then shortly after, the disease was introduced as well. So we didn't have it here in California. We have a massive citrus industry. The vast majority of our industry is fresh fruit. And so this disease poses a very great threat to that citrus industry. HLB has already wiped out thousands of acres of citrus in Florida, and they're not really sure if they're going to be able to rebuild the industry there, simply because this is such a nasty disease. It destroys the fruit, it reduces the tree, it stunts the tree, it does a whole bunch of stuff that we don't want. Currently, the disease itself is only in existence in very Southern California, so below the grapevine. But with that said, all it's going to take is the movement up across the grapevine, and then it's just going to spread like crazy, because the Asian citrus psyllid is established in the southern part of the San Joaquin Valley. So we're all just crossing our fingers and holding our breath and hoping that it doesn't get over the grapevine, and that we figure out something in the meantime to fix it. Lots of research money is going towards this disease to try to figure out how to make things better for the citrus industry. Oh, there we go. All that good stuff in the background that I just talked about, and a few more things. So it's also unculturable, and that's something that I forgot to mention previously. Unculturable meaning that we cannot take a small piece of it. We can't get that bacterium to grow outside of the plant. And that makes it really, really difficult to study. We call this an obligate parasite because it requires the host to live. And so until we can actually culture it, until we can grow it outside the plant, it's very difficult to study control methods or management methods to try to figure out how to essentially kill it. So until that happens, we're also kind of behind the times. That said, there are ways to study this disease from different perspectives as an obligate parasite. It just makes things a lot harder. Other obligate parasites are like powdery mildews, downy mildews, rusts, and viruses. You can't culture a virus outside of a plant or outside of a human. You can clone it though. You can clone the sequence. So there are ways to mess around with it, but you can't culture it and throw things at it. You know, like I can't test an antibiotic against the causal agent of Huang Long Bing. So it makes things a bit more difficult. So as for the disease cycle of Huang Long Bing, we have citrus and Asian citrus psyllid as the vector. We have citrus as the host, and we have Candidatus Liberibacter asiaticus as the causal agent. And in this case, we have a vector. So there's also a whole nother system associated to, you know, where the pathogen or the organism resides inside the vector. Does the pathogen reside just in the feeding tube? Does it reside in the stomach? Does it reside in the gut? And there's so much more to that. There's a whole realm of vector studies to understand how these things even just survive in the psyllid, or how these things survive in the insect vector, instead of just being let go at the end of the cycle where, you know, normally things would be digested and, you know, yeah. So currently, how are we managing Huang Long Bing in California? Well, quarantine's number one. So there are a number of counties and a number of parts in California, you know, where you have to, that plant material has to go through inspection to pass on to that next segment or region of land. And this is they're checking for psyllids. They're checking for symptoms related to HLB. So quarantining is one way. Pesticides to reduce the Asian citrus psyllid populations, to reduce vector populations. infected tree removal, otherwise known as roguing. So removing the inoculum source, getting the HLB that is there out. So this also consists of, sometimes knocking on someone's door and saying, hey, that orange tree in your yard is showing symptoms of a disease that could be very devastating to the citrus industry. Is it okay if we take samples, and if it comes back positive, we'll have to remove it? So there's a whole system there as well. And then, of course, planting Huanglongbing-free material, so material that's been tested for HLB. How did it get here? It originated from China, and ACP was introduced to California from Mexico. So we live in a global society. We live in a global economy. Plant material moves around often. It's constantly being checked for various things, various pests and diseases, but sometimes things get through. And this is why sometimes, like when you come off of a flight after you've been in Chile, or you've been to China, or you've been to Egypt, this is why they ask you, are you bringing any agricultural products? Have you visited any agricultural fields? Have you gone on any agricultural tours? This is why they ask you, because they're gonna ask, okay, what do you got? We need to look at it because we don't want some of the pathogens that they have, and vice versa. When we go to Chile, when we go to China, when we go to Egypt, they'll ask us the same things. Are you bringing agricultural products, etc.? Because there are things that we have here that they don't want, and they have things there that we don't want. So, exotic pests and diseases, since we're talking about introductions, things coming to places where we'd rather they not be. Exotic pests and diseases don't typically have natural enemies in their new home. So they don't have predators. They don't have things feeding on them or killing them. They evolved elsewhere. So they can be devastating. A really good example is Dutch elm disease that came from elsewhere. Glassy winged sharpshooter was introduced in the 90s, I believe. And that is an excellent vector for Pierce's disease, which is, again, a bacterial disease that has the capacity to just wipe out vineyards. Plumpox virus is a perfect example of an exotic disease. And it was introduced to America, but they were able to eradicate it. The vine mealybug is another excellent example. It just reproduces like crazy. We didn't have it here, now we do. And it's a pain to control from just the perspective of the vine mealybug, but it's also an excellent vector for certain grapevine viruses. Chestnut blight was an introduction, and it is the reason why we do not have massive stands of chestnuts throughout the eastern United States today. Wiped out trees that were, some people say, as big as the redwoods. So very sad. So diseases can spread by cankers and water splashing, wind-blown rain and wind, insects, irrigation runoff, contaminated seeds, infected transplants, soil and debris off of your boots or shoes. Again, that's why they ask you, did you tour any agricultural fields? Because you could very well bring something in just on your boots. Tools and equipment for sure, and infected plant debris. So, if you've got a black spot in your roses, we always say, okay, get all the infected leaves out, put them in the garbage, because they'll just continue to spread to your new healthy leaves. As I said before, this is prime reason number one why customs asks you all those funny questions. Are you bringing food? Are you bringing oranges? Are you bringing any agricultural products with you? Of course, they also want to know if you're bringing over illegal drugs, but that's kind of a gimme. They don't want illegal drugs coming into the country, but also we don't want exotic pests and diseases coming into the country. Remember those mystery seeds from China? This was likely an innocent mistake, and I think that it was cleared as an innocent mistake since the time I actually made this slide, which was many, many moons ago. But this could be considered bioterrorism, because if they were sending over infected seeds and someone planted them, and it turns out to be some exotic disease that could wipe out, all the squash or all the pumpkins or all the wheat or whatever, what have you, any of our staple crops, we don't have food. So it really matters. And when those seeds did show up, like we all the master gardeners got the notification like, hey, let people know, don't throw them away, don't plant them. They need to go to the CDFA. We need to find out what's going on with these seeds, and they need to be inspected. And there was a whole slew of things. And these guys just wanted to send some seeds and make some sales, but could be considered bioterrorism. So that brings me to the principles of plant disease management. And a fun little thing to remember is party. Everyone loves to party, right? So P stands for prevention, A for avoidance, R for resistance, T, therapy, E, eradication, and the next E, exclusion. And so exclusion goes right back to what I was talking about with the seeds from China. The number one line of defense is to just keep it out. If you don't have a pest or a pathogen in your region, the number one thing to do is try to keep it from getting established in your region. So the USDA Animal and Plant Health Inspection Service has a number of staff members and academics, et cetera, that just work on pest exclusion. So this is why you get stopped in Tahoe when you're coming over from Reno. That's what that big building is, where they inspect your cars, because they want to see if you're bringing plant material. And if you are, they just want to inspect it. And if it has something in it that shouldn't be here, they're going to take it. That's how it is. So please don't hide anything. Let them look, because it can be devastating. So I know that your desire to know more is intensifying. Don't worry, I got more. So as for protection, this is preventing infection by means of a toxicant or some other barrier to infection. So this is like fungicides, insecticides. Protection does wear off. So oftentimes products will have residuals, so that you'll look for like a residual, some fungicides will last up to 3 to 4 months, and some may only last 7 to 10 days. And it really depends on their mode of action. It depends on their chemical makeup. It depends on if they're systemic or if they're a contact kill. So it really just depends on the product. And it's all on the label. Label is the law. So always check the label. Avoidance. A perfect example of avoidance would be like building a raised bed in your backyard and planting vegetables in it. So there are a lot of soil-borne pathogens, Phytophthora, Armillaria, and then there's pests like nematodes, and they can be in your soil. So one way of growing your vegetables without having to deal with any of those pests is to build a nice big planter box, put a bottom on it, like fabric or even wood itself, or just build it on concrete, whatever works for you. If you are planting it on ground, you could always do chicken wire to keep the gophers out. This is a perfect example of just avoiding the problem. And you've literally put your garden in a place, with soil. You're going to go get the potting soil and the clean topsoil from a hardware store or from a garden center. You're going to put that in there. So you're just literally avoiding the issue. Resistance is an excellent way of dealing with plant pathogens. This is utilizing cultivars that are resistant to or tolerant to infection. So there are tomato varieties, for example, with the code VFN, and this means that they are resistant to root knot nematode, Verticillium, and Fusarium. So you can actually seek out resistant cultivars to help you. Also, there's a ton of research and development in developing future cultivars for resistance. So if you want to do that, that's something you can do. The therapy is a good example of this is like heat therapy. So there's actually a facility through UC Davis on the campus, but kind of off campus. It's on Hopkins Road. They do a virus therapy, and they do it with strawberries, grapes, sweet potatoes, and a handful of other crops as well. And what they're doing here are a couple of different types of therapy. One of them is heat therapy. So kind of like how when you get sick, you get really hot, you get a fever. So say you get the flu and you get the fever that's associated with that. Your body's fighting off the virus. And there are theories out there that that heat is somehow associated to actually reducing virus load. And there are some experimental evidence. There is some experimental evidence of that. And some of that is in grapes and strawberries. So if you expose virus-infected plant material to rather warm temperatures where the plants can still survive, so we're talking about like 110, 115 degrees, the virus, the amount of virus will be reduced. So that's one thing, we call it heat therapy. So you reduce the virus load to, nothing, and then you tissue culture that. Another form of therapy that they do at Foundation Plant Services through UC Davis is micro shoot tip tissue culture. And this is where they are literally taking a very, very small piece of meristematic tissue, and that meristematic tissue is underdeveloped enough that you can clone it, but it's not developed into vascular tissue yet. So most of the viruses haven't been able to move into that tissue. So you can actually clone that little piece, and we're talking 0.5 millimeter. I've done it. It's under microscopes. It's very tedious, but it's so incredible. And you literally clone that material into a tube and you grow it out. And then you test it later for the viruses that you're interested in to make sure that they're not there. But that's another form of in this case, virus therapy, heat therapy, tissue culture therapy, that sort of thing. Eradication is another form. Really excellent example. This is the European grapevine moth, which was introduced to California and was eradicated. This is to eliminate, destroy, or inactivate the inoculum. Another excellent example is plum pox. As I was stating before, the USDA did declare the United States free from plum pox. It was introduced many moons ago, and it could have taken off and it would have been nasty, but it didn't. They were able to get infected material out and burn it and get rid of it before it really took hold and got established in this country. That would have destroyed the fruit industry easily. And as we stated before, exclusion. So again, the European and Mediterranean Plant Protection Organization and the Animal and Plant Health Inspection Service are both excellent forms of exclusion. So they're checking that material when it comes into port. This is part of the reason why sometimes produce might sit at port for a day or two, because it's going through inspections. And if there's a lot of inspections, it takes time. So a perfect example. So let's talk about hot topics. I think it's important, and I think plant pathology is an excellent opportunity to talk about these different hot topics like genetically modified organisms, pesticides, and also viruses. So crown gall. So I'm a walnut advisor. So I work with walnuts, apricots, cherries, and grapes. Crown gall is a problem in all of those crops. Crown gall is a problem in many crops. In grape, it's a different species, but we're not going to talk about that today. But crown gall is in particular a huge problem in walnut crops. And we're actually looking at crown gall in this picture. It's that weird black growth. It's kind of craggy. It's kind of almost rocky looking at the base of the tree. Walnuts are hugely susceptible to crown gall. Paradox is one of the rootstocks that we graft English walnut onto. And Paradox, that's just a hybrid between English walnut and Northern California black walnut. But Paradox is highly susceptible to crown gall. And so when you're driving down the road, say you're driving down I-5 or 99 or wherever you are, and you pass by a walnut orchard, you might see some of this as you're driving by because it's just that obvious from the road, unfortunately. And what it does is it actually girdles the tree. And the fascinating thing, so it's a huge problem, right? And we want to study it and we want to figure out how do we control crown gall. So what do? We start looking at the pathogen. Luckily, the pathogen is culturable. So we do what we call plating. So we'll take a sample of that material and then we place it on some agar and grow it out. So it's culturable. But scientists were researching Agrobacterium Tumofations, which is the causal agent for crown gall. They were researching it back in the 80s. And they found, fascinatingly enough, that this bacterium is actually genetically modifying the plant tissue. And it's doing it via a plasmid. So the plasmid is this circular, you can see the bacterial cell to the left. You see the bacterial chromosome. So it's all of its genetic material for the bacterium. But then you see this thing called a tDNA or a TI plasmid. Now, TI plasmid, we're not too creative as scientists. That just means tumor inducing. And that relates back to that big giant gall that we saw on that tree. We just call it a tumor. And it essentially is a tumor. So the TI plasmid, tumor inducing plasmid, is this plasmid that contains genetic material that allows the bacterium to insert, so that plasmid will be inserted into the plant's DNA. And now the plant is basically making food for the bacterium. So on that TI plasmid is also genetic sequences for opines and hormones. Because the bacterium is just like, hey, I'm hungry, and this is how I do things. All right, so I'm moving in. You're gonna make food for me. You're my new food factory. Welcome to my life. And now the plant is producing these opines and hormones. Unfortunately, now the plant sees those opines and hormones, and they look very similar in chemical structure to growth regulators. So the plant produces callus tissue in response to the presence of those opines and hormones. What's really neat is that now we know how you can modify genetically plants. So scientists were able to pull that plasmid out, and they cloned it. And then they were like, well, we don't need the opines and hormones, but we'll put something else in of interest, and futuristically speaking, it could be a resistance gene to Fusarium. Futuristically speaking, it could be a resistance gene to Claviceps purpurea, which can kill people. So from that perspective, it was like, wow, this is amazing. Scientists can develop this and utilize it to genetically modify plants to do things that we want them to do that would benefit us from one perspective or another. They actually use this technology to develop crown gall resistant rootstocks for walnuts. Unfortunately, they never made it to a commercial level because by the time they were available, everyone hated GMOs. So there was no industry interest. But you can say, beyond the benefit of a doubt, that GMOs are derived from nature. They are natural. So let's move on to the next interesting pathosystem, Bacillus Thuringiensis. So Bacillus Thuringiensis is actually bacteria, and it is an OMRI certified pesticide. OMRI basically meaning that it's organic, it's organically approved. It's an Organic Materials Review Institute is what OMRI stands for. So BT is what it's commonly called. It's in Monterey pesticide. And yes, there are pesticides in organic farming. But Bacillus thuringiensis produces a protein that kills caterpillars and worms, but not humans. So it's activated when it enters the gut of the insect. It does nothing inside of our bodies. Fusarium ear rot of corn is caused by Fusarium verticilloides and a few other Fusarium species. This fungus produces fumonisins, which again are mycotoxins. We talked about that with ergot of rye. These mycotoxins are harmful to livestock. They are potentially harmful to humans, and they infect wounds made by insect feeding. So you could essentially figure out how to genetically create that protein, right? And you could actually modify plants to secrete that protein that's produced by Bacillus thuringiensis. And they did. That's what BT corn is. This reduces insect feeding, which reduces Fusarium ear rot, which reduces fumonisin toxins, which leads to healthy corn and healthy horses, right? Can't be too upset about that. So what's a pesticide? Well, according to the Department of Pesticide Regulations, a pesticide refers to not only insecticides, but many other kinds of chemicals. Under state and federal law, a pesticide is any substance intended to control, destroy, repel, or attract a pest. So essentially, a pesticide is anything that controls a pest, right? So technically, if if insects are a pest, insects are a pest, these worms are a pest. If BT controls those insects, what does that make BT? Well, that makes BT a pesticide, right? According to the Department of Pesticide Regulations, their definition, BT is a pesticide. It's an organic pesticide, sure. And if we genetically modify corn to contain a protein associated to killing pests, well, I suppose you could probably spin that one way and you could say they put pesticides in our food. But I would argue that that's a very painful misinterpretation of the truth based on what I've just said. So let's move forward to tobacco mosaic virus. Actually, sorry, we're going back in time now. This work was done even before BT, or maybe around the same time, actually, now that I think about it, the 80s. A lot of great things happened in the 80s, not just really great music. So tobacco mosaic virus, they're studying this virus. The virus is a problem in tobacco. It's been a problem for ages. It's easily transmissible. You can actually move it from a cigarette to a tobacco field. You can also move it into tomatoes. You can move it into other solanaceous crops. So it's a pain in the butt. There was a lot of really cool research going on at the time, and they were able to genetically modify tobacco plants to express a coat protein that was a part of the tobacco mosaic virus. And in so doing, they eliminated the symptoms, created virus resistance. So these tobacco plants didn't become consistently infected by tobacco mosaic virus, and they didn't show symptoms. That was really cool. Then we started seeing issues with papaya ringspot virus in papaya. This is a healthy papaya. Papaya ringspot virus had moved into Hawaii, and that was a big industry, big papaya industry. It was decimating crops. It started wiping things out in about 5 years. And it can also infect cucumbers and other cucurbits. It's spread by aphids. So it's a podi virus. It's spread by aphids, and the aphids pick it up very quickly and transmit it very quickly. And aphids are extremely common. If you've ever had a rose bush, if you've ever had anything, you've had aphids. So the aphid comes in, it feeds off of on an infected plant, and then just moves right over to the next papaya plant and infects it. And this happens in seconds. So what does that mean? That means that controlling the vector is pretty much irrelevant. It's a waste of time. It's a waste of money. It's a waste of pesticides. Like, there's just unnecessary. It's not going to do anything. So Dr. Dennis Gonsalves and his team decide to embark on genetically modifying papaya to produce the capsid protein of the virus, of papaya ring spot virus, because in tobacco mosaic virus, it was shown to work. And they're like, well, let's try it in papaya, see what happens. And in 1998, rainbow and sunup seeds are released to growers. And as you can tell by the photos, you have in the black photo, you have on the left the rainbow papaya, and then on the right, you have the really sad, sick, infected conventional papaya. And then in that lower photo, below the black photo, you have on the inverse. So on the left, you have really sad, sick papaya. And then on the right, you have the rainbow papaya. So the interesting thing about this is that this work, utilizing the capsid protein or the coat protein to derive resistance to viruses, was developed way back in the 80s. So interestingly enough, with the COVID-19 vaccine, although the system is very different, don't get me wrong, the science is still similar in the sense that they're utilizing, they sequenced the virus, and then they sequenced, and I have a fly, sorry, buzzing around. They sequenced the mRNA sequence that would code for a spike protein, which is on the capsid or on the coat of the virus. codes for a spike protein so that when you're injected with the mRNA, the spike protein is produced inside your body. And then your body can build antibodies because your body then reacts to the presence of that protein and builds antibodies to fight that presence. And that antibody is specific to that spike protein. It's specific to that shape. So now your body is ready in case it gets infected in the future with COVID-19. So thinking about timelines, this technology is actually older than your smartphone when you really think about how long this work has been done and been developed and discovered, for that matter. And we really owe a lot of it to the invention of PCR, which was way back in 1983 with Kari, who later won the Nobel Prize. So I think it's really incredible. It's hard to live through what we're living through right now, but it's also incredible from a scientific perspective to see how quickly we can move through it. So with that, looking forward, if you want to become a plant pathologist, there is so much room and opportunity for you because 10 to 15% of crop loss in the world is related to plant disease. And so we really need future outlooks for food security. Number one, there are still people starving in the world because of plant diseases. And that's really unfortunate because, you know, we can help. We can tackle these challenges and we can figure out how to make sure that people are getting fed and able to grow their own food. Oftentimes in many places, you know, they just don't know what to use or what they need isn't available to them to be able to grow food. We have to think globally because, you know, as we just discussed, things can be introduced, et cetera, but also we're very much a global economy. And so we have to think about everyone in that system and how to work with it. Climate change is huge. You know, recent estimates say that certain cities could be underwater by 2050, and that not like submerged, but where they have some water where they shouldn't. And that's really something that we need to take into consideration looking forward. How does that impact plant pathology? Does it increase or decrease certain pathogens? Does it allow for other pathogens to become bigger issues than they were before? You know, this changing of the environment, how does that impact how our food is grown in general, but also as it gets hotter and hotter, there are certain products we can't use when it's really warm to control certain pathogens. And there are certainly some pathogens out there that like it warm. So changing the climate means that there's going to be a change in all the systems that we know and understand today. And so keeping an eye on that and knowing where we're headed and how to deal with that, it's huge. There are many challenges posed to conventional ag, and I'm not saying that organic is better. I'm just saying that, you know, a lot of products, a lot of pesticides are being regulated or removed, and that's making it harder and harder. Certain pesticides are becoming ineffective simply because there's only one or two options. A big kicker to using fungicides, say if you're controlling powdery mildew, a big problem is being able to stop resistance from forming in the population. And so you have to change your chemicals, you have to rotate them and based on their mode of action so that you're not constantly selecting for a population that's resistant to say one or two chemicals. And some of these products, some of these pesticides, some of these fungicides, they may not be effective at certain temperatures. They're limited. Some of them are just limited in modes of action. So there's a limit as to which types of products will even be effective on certain pests and diseases. And that's just the tip of the iceberg. There are a lot of other reasons that, you know, conventional ag has a lot of challenges being posed to them. And there aren't a lot of easy answers. This is not a simple equation. You can't just put compost on it and walk away. It's not that easy. There are downsides to compost. Sometimes compost has certain elements in it that you don't want building up in your soil. So there's no easy answer, and it's really up to us and the future generation to know how to deal with these problems before they become decimating. And there's always technology advancing, which is so great. We move as technology moves, we advance as technology advances. 30, 40, 50 years ago, they didn't have the machinery that we have today to harvest corn and to harvest apples, and et cetera. So technology continues to advance, and we need to stay either with that or ahead of it, or be a part of it. There's opportunities for engineers, there's opportunities for people in mathematics to write formulas and models on temperature and climate and everything else, and how does that apply to fruit and productivity and et cetera. So there's so much. Speaking of advancing technology from an agricultural perspective, mating disruption is big. Certainly if it works, you can really confuse a male from finding a female, and then you can disrupt mating. So you can reduce the population of an insect from the perspective of mating disruption. That does require for an insect to be producing a pheromone as an attractant. Some insects don't do that. Some insects actually vibrate the plant, and that is like the call. That's the, hey, I'm over here and I'm ready, is vibrating something. This is just crazy to think about, but how do you confuse something? You just put something on the vine to consistently vibrate the vine? I don't know. That's the future of ag. Marker assisted plant breeding, being able to use PCR and look for genes that are desirable, that we want to save, instead of having to put 100,000 seeds out into the field and wait 3 years to see what emerges as your good plant versus your bad plant. So marker assisted breeding is big. CRISPR, CRISPR is becoming sort of the current and future of being able to add or subtract things. Say the plant reacts to a pathogen, a certain gene is triggered. Well, what if we just alter it just slightly so that it doesn't recognize that pathogen, and then all of a sudden it doesn't go into programmed cell death, and all of a sudden you've just created a tolerance, and that's not even, maybe you took out one nucleotide or something. It's very minimal. So CRISPR is very big, and there's a lot of future work in CRISPR. Also, a sterile insect technique. This worked really well for the pink bollworm in cotton. And if you want to know more, feel free to look it up, because it's a fascinating story. But, just introducing a lot of sterile males so that they just kind of take over the population, and they can't mate with a female, but, the female thinks it's mated, and then it goes on with its generational changes and dies without creating a next population of problematic insects. So tons of futuristic ideas and research and current events going on. So I think with that, I will ask for questions.
Hey, Kari, that was awesome. That was a great talk. I loved it. And I think there's a lot there for my botany class. I have several students that I know are in a couple other classes today, because I sent them a little message and they quickly got back to me. Hey, I'm in class. Can I watch this later?
Oh, cool.
And I said, yes, but I did want to ask you, are you okay with us keeping today's talk on our MJC Science Colloquium channel?
Yes, absolutely.
Awesome. Good.
If you want it up there.
Yeah, that's great. I'll use it in the future for other classes also. Hey, I'm just curious, the mystery seeds, do we know what they were now?
I think they were like, I think some of them were squash and pumpkin.
Okay.
I vaguely remember, but the seeds, the picture that I had there, those look like pumpkin. And now I think I pulled that.
Okay. I was wondering if we found out. I remember the story and I was like,
yeah.
What is going on? That's so weird. I'm like,
yeah, it was a couple of different types of seeds. I remember that. And then it was just a ploy. It was a marketing ploy to try to get to people to buy their seeds. And it's like, but you can't do that. So that's illegal.
So weird. One question, pomegranates. I have that leaf-footed bug.
Oh, yeah.
Yeah, problem. It's become a huge problem this year. Do they also act as a vector for other diseases? Or
Gosh, that's a good question. I would have to look that up. Speaking to that, number one, pomegranate, apparently leaf-footed bugs love pomegranate.
They do.
I'd have to look it up to see if leaf-footed bug is a vector of anything of dire concern. But I do know that we actually have we have an integrated pest management advisor, Jalenda Rajal, at our office in Stanislaus. And he's got a few studies going with leaf-footed bug. So
okay,
yeah,
well,
I do.
he wants to come and sample my pomegranate and so it's really close to my planter boxes. And so my tomatoes, they hop back and forth, and I've tried soaps and other things. And it's really bad this year.
Yeah, well, and so it's been interesting. Some insects have been worse this year. Some have been better. And it may or may not be related to, a drier winter and that research may even exist, but with a lot of this stuff, some of this is just completely unknown.
Okay, who was it again that's working on that? Because I do want to get more info.
Jalenda Rajal. I'm more than happy to introduce you via email. So,
that'd be great. Yeah, that'd be
shoot me an email.
And then I think and then just for the benefit of my students, when you talked about crown gall, what does it mean to girdle a tree?
Oh, very good question. A really good parallel would be like if you were to just tie a rope around a tree and then the tree slowly grows around that rope, you're girdling the tree. What's happening is you're restricting the vascular tissue. So what happens with crown gall is these callus galls grow and die on an annual cycle. So they grow and then they crisp up and die and fall off, and you get these big gaps in the vascular tissue. So you're essentially girdling the tree.
So basically the tree's starving because the nutrients aren't coming back down through the phloem to the root.
Certainly if that gall, if enough of it grows all the way around, or even about 3/4 of the way, the tree just kind of declines. If it shows up in later years, it's usually manageable. If it's there in the first couple of years of the orchard establishment, you practically have to replant. If there's a lot of it, if every tree or 75% of the trees are infected, growers will just remove the trees. But I could spend an hour talking about crown gall if you ever want me to. So a lot of it comes down to sanitation. A lot of it comes down to sanitation. Something I didn't mention was, you can spread it with dirty pruners. So you can pick it up from the soil. It's ubiquitous in most situations. So you can pick it up from the soil. And if you don't clean your pruners every time you go to, do a new pruning, you may end up transmitting it to that plant because it enters through wounds. So oftentimes you'll get it, where there's like suckers that have been cut back, or a dirty pruning job. But there are also other ways that it can be introduced to the system, which would also make just a really great talk. So
great. Hey, I just also wanted to say that I thought you made an excellent point on the age of the technologies behind the SARS-CoV-2 vaccine platforms and showing how that's science kind of goes across. Yeah, I love that. So thank you for that.
We both did.
Yeah.
I wasn't sure it would be too controversial, but I went for it.
I don't think it's controversial. I think it's sound science, and it's a good way to show the relationship. Yeah. And then Denise, do you have any questions today?
No, a lot of the stuff I've already covered with Kari and some of her other talks, so I'm familiar with a lot of it. Yeah, and some of the research that she was talking about during the little cells under the microscope to make the clones, been there and done that.
You did it too. Yeah. Oh, it's fun. It's a little tedious after a while.
Good memory.
Really cool.
Yeah, it was a great talk. It was.
Thank you. Thank you.
Well, that was terrific today, and I really appreciate it. I think we might have to have you back again, maybe to talk even more specifically about some other plant diseases. And
Absolutely.
I really appreciate your time today being with us.
Your passion for this is very obvious, Kari. Yeah.
Oh, thank you. Thank you.
And I like that we are talking to students and you're giving them ideas for future careers in a lot of different areas. Yeah.
We definitely need more plant pathologists. Yeah.
Sure.
Yeah. Well, thank you for today. And I guess we'll sign out for today. And I hope everybody will join us in a couple weeks for Greg Stock, Yosemite National Park geologist, joining us to talk about glaciers. Hope you guys have a great afternoon. Bye.
Thank you, everyone.
Thank you, Kari. Bye-bye.
Thank you.
Bye.